Hi, my name's Amy Holmes. This is my Unit 3 DNA, a new way to translate DNA beyond genetic code presentation. This review is from a 2019 by Giorgio Bernardi in Bioessays entitled The Genomic Code of Pervasive Encoding Molding of Chromatin Structures and a Solution of Non-Coding DNA Mystery. Non-coding DNA sequences do not code for an amino acid. Most non-coding DNA lie between genes on the chromosome and had no known function at one time, while others, called introns, could be found within genes and play a role in the regulation of gene expression, such as how much a certain gene is turned on or where they should turn off, how the DNA is packed into the chromosome. When non-coding DNA was first discovered, there were three main explanations as to why they existed. First one was they were pseudogenes or junk genes with no purpose. They served no means of anything. They were just there and existed. Second was the selfish DNA, which was termed based on them being a molecular parasite to their host, with no known function, just there and basically sustaining off of the host that they were attached to. Junk and selfish DNA were found to be about 80 to 90% of the genes that were found in the DNA. The third was the majority at 80% of genomes took part in at least one biochemical RNA and or chromatin associated event in at least one of the cell types. However, with the discovery of isochories' involvement in the formation of chromatin domains and spatial compartments, it was discovered that there was a correlation with isochories and their location as far as an expression of regulation of the gene. Bernardi's paper looked at that and talks about non-coding DNA helping with the folding of the 3D structure of DNA. And this was evident by looking at long strands, 100,000 base pairs long, of DNA with unique structure called isochories that can be seen and are defined. They're defined in their location or association with their GC content. There's two forms. There's the LAD, the lamina associated domains, and they have low GC content. And the second is the TAD, or the topographically associated domains with high GC content. These are associated, and the TADs are associated with peaks that are created in the DNA, and they have open chromatin structures. Bernardis hypothesized that these open structures helped with transcription of the genes by allowing them to be more accessible to proteins and enzymes within the nucleus. My personal opinion on this paper was that based on the information that Bernardi presented, he made sound argument that non-coding DNA plays a significant role in the formation of the 3D structure. Without that non-coding DNA, the 3D structure might not present or be as effective or express itself in the appropriate manner. And that's huge, it kind of reminds me of the human body. We have bones bones are very basic and could at one time probably were thought they're just there to hold up the structure, which is basically what the non-coding does in helping with the role of the DNA structure. This concludes my Unit 3 presentation.